Great. Well, I'd like to start by thanking Dwight for inviting me to give this little presentation for y'all and to all of you watching this. So what I'm going to do today is tell you a little bit about my career. I'll start obviously with this slide of what my career is, and then tell you a little bit about how I got there. And then I'll tell you about some of the research that's happening in my lab. So currently I'm an associate professor here at the University of Texas at Austin. I'm actually up for promotion, so hopefully next year that will be full professor. I've been here since 2008. And it's funny whenever you're out and telling people what you do and you say that you're a professor, the first question you always get is, what do you teach? And so one of the things that people get surprised at is, like, at a research intensive university, I do teach, but I don't teach that much. I have one class that I teach. It's developmental biology. That's the research that my lab does. That's what I'll be telling you about today. And so I really love teaching, and I've actually gotten a few teaching awards from student nominations. I'm very proud of that. But the main thrust of my career is not the teaching of undergraduates in any formal classroom setting, but it's actually training scientists. So my teaching is how to perform high quality, rigorous research. And so that's what my job is most about. And it's a great job because I just get to do science, and science is fun, and I get to train people in how to do science. And it's a wonderful job because there's a really diverse group of people that I get to interact with. I get to interact sometimes even with high school students, but a lot of undergraduates. So you typically have 10 to 15 undergraduates who will be working in the lab. And then graduate students, typically around 5 of those in the lab. And then especially in science, even after you go and you to graduate school, you get your Ph.D., you're really not done training. There's a postdoc. And so where you train then to the point where you can run your own lab. And so you usually have about 5 of those. And so it's this wonderful group of really young, bright-eyed, bushy-tailed, brand new undergraduates to more seasoned researchers in that postdoctoral phase of their career. And so it's a really fun thing to do. And then the other big thing that we have to do is publish. Science doesn't mean anything if nobody knows that you've done it, right? And so the great thing about science is that we're learning new things all the time. And so we have to get that out there. And so the way we publish it, we'll submit it for publication to a journal. And then it's reviewed by other scientists who will then make suggestions or demands sometimes on how it should be changed to make it more rigorous. And this is really how science works. So we try to maintain the most rigor in the lab as possible. And then before it gets published, before it gets put out there for the community in general, or our society in general, it gets reviewed by other people to see if there are ways that it needs to be even more rigorous. And so this is the really fun part about all the science. And so while the University of Texas pays me to do these things, they don't actually pay for the research that's going on in the lab. And so to actually maintain the research that I'm going to tell you about a little bit in a little bit, I have to write grants. And so basically these are proposals with what our ideas are, what sort of research we would like to do. And then again, that gets reviewed by other individuals, and they get to decide whether it's meritorious enough for them to provide money. And there are lots of different granting institutes. The research that I'm going to tell you about in my lab is funded by the National Institutes of Health. So because it's relevant to human health. And as I go through this career portion of the talk, you'll see various grants and fellowships, and they have a little letter and a number. This R stands for a research award. So these are, fairly large grants that can fund a lab, both the personnel and the cost to just do the science. And so these are a couple of them that have funded my lab. This R01, this is sort of a standard grant that people doing research, NIH research will have. This R35 is actually supports a lab itself. Instead of an idea, you can do whatever you want with this money here. One of the things that I found when I started my lab that I hadn't really thought of. I love science. Thinking about science is fun. There's no problem there. But then once you're running your own lab, there's a whole issue where you become a manager. And it was a surprise, I would have to say, because nowhere in my training was I ever trained to manage people, right? I had people I was mentoring, but really running the nuts and bolts, you have to become an accountant when you're managing funds for a research award, and just all of the details there. And so this is actually a sizable amount of my time. And the irony is that you train to do science, not management, but then you end up managing a lot. So how did I get here? And obviously I'm giving this talk because I got here through Bethel. And so these are my dates that I attended. I was on the 5-year plan there, and I got a dual degree in biology and psychology. I wanted to back up a little bit before Bethel because I think it's really important as I reflect upon my career. I've found that, the people that you know and interact with really matter an enormous amount in your life. And sometimes it's hard to even understand, at the moment. But so I wanted to point out 2 people who actually had an enormous influence on my life and getting me to Bethel. So my 6th grade teacher here, who thought that instead of just the disruptive student in her classroom, which I certainly was, but that maybe I wasn't just a disruptive student, that maybe I wasn't being challenged enough. And so she actually got me into a gifted ed program. And so then my high school gifted ed teacher, Lila Bartel, was actually a Bethel graduate. And so I grew up in Topeka, Kansas. And I didn't, my family, so I'm a 1st generation college graduate student. College wasn't discussed in my home. College wasn't an expectation for me, but Lila was a Bethel graduate and she encouraged me to look at Bethel when I was starting to think about colleges. I was more interested in leaving Kansas actually, but she said I should check it out. And so I did. And when I met the professors there, I thought it seemed really amazing, and that they interacted with me really easily and they were friendly, approachable. And so in the end, I decided to stay in Kansas. And that's where I got to meet, work with Dwight Crable and Wayne Weens, my major psychology and biology professors. And I think one of the best things about Bethel, and especially Dwight and Wayne in particular, was the emphasis on sort of how you do science, that it wasn't simply sort of a regurgitation of knowledge that they expected. They really expected that you understood how science worked, how we got to know what we know. And that's something that I've always tried to carry forward with me. And they really encouraged research, and that led me to an undergraduate research program at the National Institute of Health, where I worked on learning and memory with Dr. Marie Webster, working in monkeys. And so this was my first real experience of NIH level research. And so I think that the thing that I take also from Bethel is really the value of a liberal arts education. I've been at meetings where students will ask, how do I think outside of the box? And the answer is to that is kind of, you have to think outside the box. But a liberal arts education, I think, encourages that, it nurtures that, because it's a broad education, and that the goal is to get you thinking, instead of just being narrowly focused on something. The breadth, I think, really has a strong value in education, and I really encourage students to pursue liberal arts education whenever I have the chance. So at the end of Bethel, I had this taste of research, but I wasn't sure what I wanted to do with my life. And so my trajectory is a little non-traditional. So I went from my bachelor's to a master's, because I sort of liked the research. I wasn't sure if it was really for me. So I just went a little south to Wichita State University and worked with Dr. Schmidt, looking at cell adhesion in the developing brain. And so that gave me my very first experience of teaching. I was a teaching assistant for a general biology class. I really enjoyed it. But I think the important thing at WSU, in addition to meeting my wife, this photo here is me at my wedding party, is that I found out that I really did like research, and I wanted to continue it. And so the non-traditional part of it is that I went from a full master's program at one university to a full PhD program at a different university. So I moved eastward to Missouri, where I got my PhD in biological sciences. So I chose Missouri because my interest that I had culminated through Bethel, and then my master's was really in developmental biology. And that's what I'm going to be telling you about, is the question of how you go from a single fertilized egg to a complex organism is just fascinating to me. And so Columbia was hiring 2 brand new developmental biologists, and so that really excited me. So I went there and I worked with one of them, Kathy Kroll, and it was a great experience here too, because she was just setting up her lab. I was her first graduate student. I got to see how a lab got started, and it was really a complete immersion in science. Kathy was passionate that all of her students interact with the broad scientific community. We went to meetings, met eminent scientists. It was really an amazing time, and I had a great view out of my window here. Here's where I really got started mentoring people. Kathy was really, she wanted all of her grad students to mentor undergraduates. And I really started honing my scientific communication skills. So she was also felt that her graduate students and postdocs should present at scientific meetings as frequently as possible. And so this is where I really got started there. I continued my teaching. I got a TA award at Columbia. And this is where I got my first inkling of what NIH funding was like. So again, a letter and a number. This T means it's a training grant. So this is a grant that is given to the university, so Columbia, and this is for doctoral students. And then the university picks those students that they think are meritorious for the award. And so I was provided one of these by the university. So then, as I mentioned, typically in the sciences, you would go from a PhD to a postdoc. And so my work with Kathy Kroll, we were looking at axon guidance, so how the central nervous system and peripheral nervous system gets wired up. How do neurons find their targets? And we were doing this in chicken embryos. And so typically what we would do is some sort of scientific manipulation, and then we would fix that embryo up and section it. And what I really wanted to do, again, this idea of development being fascinating, I wanted to actually watch development happening in a live embryo, not in a fixed one. And so to do that, I joined the lab of a zebrafish researcher. So I went from a brand new researcher to perhaps the most established researcher, zebrafish researcher on the planet, Dr. Chuck Kimmel at the University of Oregon. And so I chose zebrafish, and you'll see some of these movies here in just a little bit, because you can actually watch the embryo develop. They're small, they are transparent almost. And so you can image the cells within the embryo in the living embryo, and you can also manipulate them genetically. So it had all of the scientific tools that I was interested in. And so in Chuck's lab, not only did I learn how to do zebrafish work, but he was really keen on written communication, in addition to oral communication. But so he really hammered me very hard on being able to write well. And it's a very important skill for a scientist. Again, as I said, if the science doesn't get out to the community, you might as well not have done the science. And so both writing manuscripts and then navigating the NIH grants. They're complex. It's not going to be the focus of this talk, but then there's this F, and so this is an individual fellowship. So this was awarded by NIH to me to train. It provides just salary, basically. And then this one was important in my career. It's called a career grant. I was among the first awardees of this K99R00 award, where it funds the end of your postdoc with this, and then the beginning of your independent career with that. And so then from there, I was able to get my job here at UT, where we do the research that I'm going to tell you about now. And so most of the research in my lab focuses on the development of the head. How do you make this structure on your shoulders? And a lot of it is on the hard tissue. So how are you making your skull? And so most of your skull, or the skull of this noble creature here, is made of a cell type that we'll be talking about a lot today, called neural crest cells. So these are a vertebrate specific cell type that were essential for the evolution of the vertebrate head. They make a number of different tissues, including the bony tissues, the connective tissue, melanocytes, sensory organs, peripheral nervous system, among a few of those tissues. So we're really interested, a large portion of the lab is really interested in how you make a particular shape. And not only how you make that shape, if your skull is even shaped correctly but not interconnected appropriately, it's not very functional. So how do you make this shape but then also integrate sensory organs, the brain inside there, and connect it to a muscular system to allow it to move? And so these are some of the things that we do in the lab. I'm not going to go through them. And for time's sake, the only one that I have time to talk about today, I'm going to tell you a little bit about our work on gene-environment interactions. A large portion of the lab works on this question itself. And so I'll give you a little bit of an introduction to craniofacial development and the idea of gene-environment interactions, and in particular, the environmental exposure that we're looking at, ethanol, and then we'll get into some of the data from the lab. So as I said, the vast majority of the craniofacial skeleton is derived from neural crest cells. So this is a cell type that is born in the neural tube, which is going to generate your central nervous system. And then because they make all of these different tissues that aren't in your brain or your spinal cord, they have to leave that neural tube and migrate away into the periphery. And so here is a schematic of a zebrafish. This is another reason that we like to use them. So this is 12 hours post-fertilization. And so by 12 hours, the fish has an eye, it has what's forming its mouth, and these little colored arrows here depict streams of neural crest cells that are migrating in the head to make the craniofacial skeleton. And this is a live zebrafish. This is why I work on zebrafish right now. And so this is its eye. And what we're looking at, these green cells are all neural crest cells that are labeled transgenically. And we're looking at this stream here. So they're migrating away from the neural tube. And you can see some here coalescing around the mouth that are going to make the jaw and the palatal skeleton. So by the end of the first day of development, these neural crest cells have populated these structures, which are called pharyngeal arches. And so here is another transgenic zebrafish. This is the eye here. We were zoomed in on this little region previously, so now we're zoomed out a little bit more. But after they migrate, these neural crest cells interact with other tissues. That oral ectoderm, or this mouth that's forming that you see there. And then here, this fish also has a is transgenic to label the endoderm, which is this red. And you can see all this morphogenesis happening, these structures starting to form. And so it's these interactions across these tissues that are necessary then to form a shape of a skeleton. And so this happens in zebrafish across 4 days. And so we can visualize development very rapidly. This is why we like to use this. And so that structure looks like this. So these are zebrafish embryo or larvae that have been stained for cartilage in blue with Alcian blue and bone in red with alizarin red. There are lots of different structures. You don't need to know what any of them are called, even though they're labeled here. But here, this view we'll see a bit where it's a dorsal view. We're looking down. So this structure sits immediately below the brain and above the mouth. So this is analogous to your palatal skeleton right here. And if you flip this fish over, there are these reiterated cartilages. And if you pluck some of them out and put them sideways so that you can take a look, you can see that they all have a very specific shape. And so when we look at our gene environment interactions, we're really looking to see if any of those alter the structure or the shape of any of these elements. And so then, as I said, we're using the zebrafish as a model for human birth defects, not because we care a lot about zebrafish birth defects. And so the reason we can do that is because the genes that control the development of the fish's face are by and large the exact same genes that control the development of your face. And so these details here are not so important. These are just genes that are known to be involved in human craniofacial development. And Mary's just showing that they're expressed in zebrafish in similar places. Again, the details here aren't so important, but here you're looking at neural crest cells that are going to generate the palatal skeleton here and there. And so the difference between humans, the shape of the face of the humans and fish isn't the genes that they express, but just subtleties in how they deploy those same genes. And so what we find in zebrafish is highly relevant to what's happening in humans. So now I'll tell you just a little bit about the concept of gene-environment interactions, and then we'll get into the detail. So working in a developmental biology lab, typically, most of my career has been working in a highly controlled environment where, you don't want any outside influences, but that's not the way humans develop, where in human development, there are lots of potential chemical exposures, and actually any animal that's in the wild is similarly potentially exposed. And so what then at the interface of these environmental influences is really the genetics of the developing embryo. There can be predisposing mutations that might then, when there's a particular environmental exposure, cause an effect. So gene-environment interactions are actually thought to underlie most birth defects, but they're difficult to study because you're looking at this interaction between two things. And so if we think about humans with over 20,000 genes, then those are 20,000 potentially predisposing mutations. Luckily, we're not grapes. It would become even more complicated there. But so we have 20,000 or so genes and at least 85,000 chemicals that are under the Toxic Substance Control Act. And so many of these are probably in your homes right now. And so if we think about 20,000 genes and 85,000 chemicals, that becomes extraordinarily difficult to understand the potential combinations of them. And so when I started this research at the end of my postdoc, I thought, well, let's narrow down the environmental aspect to something that's very common. And so I chose alcohol, thinking that it would be kind of easy since we know what it is. I found out I was extraordinarily naive. It's not easy, but at least we know what the environmental influence is. And so you're probably familiar with fetal alcohol syndrome, but fetal alcohol spectrum disorder is the term given to any birth defect that has a cause due to a prenatal alcohol exposure. So FAS is on the severe end of that spectrum, but FASD is actually rather common. So some estimates are even higher than 1%. In some areas, it's more like 5%. And from human work, it was clear that there was a genetic risk factor for FASD, but what those genes were really not known. And so that's where we got started. Because of the genetic tools that we have in zebrafish, we can identify these. And so what we're looking for are gene ethanol interactions where you have a particular genotype. One that I'll tell you about today is this gene, that in a particular environment, say a neutral environment, you have no defects to the face, but now you add alcohol, you actually have a facial abnormality. And so that's what we've been screening for. And we've done it a couple different ways. So one of the things that we can do is just randomly mutate genes, hitting, those 20,000 genes, and then just identify those that cause a phenotype when placed in alcohol. And so this was our first screening crew doing this forward genetics, as we call it, where we just, again, the details aren't so important. We're just randomly mutating fish, breeding them to the point where we get homozygous mutant fish, and then doing a differential exposure, one to alcohol. This is in the water. It's not the percentage in the fish. And then this doesn't cause defects to normal wild type or non-mutagenized fish. And then you compare those to control and identify ones that have phenotypes here, but not here. And then the details are we have to figure out what the mutated gene is later, since we've randomly mutagenized fish here. And so this has been quite successful. We get mutations that disrupt various regions of the face. And so these are just a few examples. I'm not going to go into the details of them. But yeah, like here you're missing the jaw. This one is extraordinarily subtle, just missing this tiny region of the face. And this one is a little bit more profound. So the interesting thing is with all of these mutants, there is no facial phenotype that we can see in the absence of alcohol. So it's only when there's an alcohol exposure that we see these defects. And then, as I said, we have to work backwards to figure out what the gene is. And so one of those we've identified as this gene here, LRP13B. It's just a member of a large family of proteins that aid in signaling between cells. And again, I'm not going to go into details, so you don't need to know more about it than that. But the interesting thing is that this gene had actually never been functionally characterized before. So it was known to exist, but no one had identified a function. And we think it's because, again, it wasn't in the right environmental condition. And it turns out that these mutants, not only are they sensitive to ethanol, but they're also sensitive to temperature. And so zebrafish like to develop at 28.5 Celsius. And here you look at the wild types, they look fairly normal, both at lower and higher temperature, but these mutants look profoundly worse at this higher temperature. So this gene is modulating various environmental exposures. And so those forward genetic approaches are ongoing, but I wanted to spend the rest of my time today talking about another approach where we can take candidate genes, so genes that we already know are involved in facial development, and analyze if they make an embryo sensitive to an ethanol exposure. And so we find that most genes that we know are involved in facial development are not ethanol sensitive. So if you provide an exposure to ethanol, it does not actually alter the phenotype. But about 20% of the mutants that we've analyzed do. So a sizable number, but still a minority. And so some of those genes we've characterized are shown up here. I'm just going to end the talk by focusing on this one, platelet-derived growth factor receptor alpha. And so I actually, as I was a postdoc, I identified this gene as being critical in development of this palatal skeleton. And it had already known to be important in palatal development in mouse embryos. And so there's an evolutionary conservation. So here you can see this midline clefting in these mutants. And I identified that defect is due to inappropriate migration. So this is that movie I already showed you. Now this is a fish that is mutant for this platelet-derived growth factor receptor alpha. You don't need to be an expert in developmental biology to see that this looks very different from that. So the embryos are at slightly different angles, but this is the eye here, and that oral ectoderm is actually way down here. And so these cells that would normally migrate to the oral ectoderm to make that palate aren't getting there in these mutants under normal conditions. But then as I was starting my lab, we did our first pathway analysis to identify mutants that might enhance ethanol sensitivity. And so this was the very first gene that we identified as being sensitizing to ethanol exposure. So again, we add a dose of alcohol that doesn't disrupt the shape of this in a normal wild type. But now if you look at these mutants where you have this clefting in an untreated fish, if you add ethanol, now that entire structure is absent. And what was more interesting to us was this effect here. So normally being heterozygous, meaning that you have one good copy and one bad copy of this gene, doesn't have a deleterious effect. But now if you add alcohol, this reveals these, and 50% to 2/3 of the embryos, we see these very striking craniofacial defects. And so this reveals what is called haploinsufficiency, where a single copy of the gene is not adequate for proper development. And we think that that's important because that's actually commonly found in humans, where a human has is heterozygous for a gene, but has some birth defect. And so we think that this is very important. And we worked with collaborators across these these various publications, and we have since shown that this interaction and that this receptor is important in human development as well. And so this phenotype actually seemed like it was due to a different cause than simply migration. And so what Neil did was to take a look at cell survival, specifically looking if there was cell death happening, this process called apoptosis, where a cell senses some influence and then dies. And so this is raw data here. The graphs show it best here down. So if you take a look, the relevant comparison here is an ethanol exposed wild type fish, so 2 good copies, versus an unexposed mutant fish, 2 bad copies, very little cell death. But then you combine these two, where you have your mutant and you have ethanol. If you were to just add these two together, you would be there, but you have vastly more cell death. So this is a synergistic effect where you combine these two things, the environmental exposure and the genotype, and you have a great exacerbation of this phenotype. And so Neil was then interested in what's happening. So Pdgfra is a receptor. So it sits on the surface of a cell and it signals into the cell. And it's a growth factor receptor. And so what that does is signal for cell survival and growth. And there's a pathway that leads from that receptor to cell survival. And this pathway involves these players. We're going to focus on mTOR here. This is a protein complex that's really critical for cell growth and survival, and was implicated in ethanol teratogenesis. And this mTOR can be inhibited by this chemical here, rapamycin. And if you put this chemical on Pdgfra mutants, you get the same phenotypes that you see with ethanol. And so this is a side view that I showed you at the beginning. So very similar phenotypes. And if you take a look at this pathway, so the way this functions is that you have phosphorylation events going from the receptor to this downstream target that's actually causing the cell to proliferate and survive. And so what Neil did was to take a look at the levels of phosphorylation of these different proteins. And again, there's a lot of data here, but the real key is that if you take a look at this very downstream target at the phosphorylation levels, they are down in an ethanol treated mutant. And so it seems like we're having an interaction through this pathway. And this is a little complex, but if you remember, I told you that Pdgfra was important for migration of neural crest cells, and then Neil's work said that it's critical for survival. And so we're really interested in now this dual function of Pdgfra. And so we're analyzing this potential downstream signaling that separates these two events. I'm going to finish just by going back and more into this mTOR complex, because as you can see here, so this little arrow means that this is activating this protein. This little hammer, as we call it, means that it's inhibiting this protein. And then you have another inhibition here. And so what we think is that the activity of this pathway modulates the sensitivity of an embryo to an environmental exposure of alcohol specifically. And so what this means, because of these inhibitory interactions, it means that we would predict that a mutation here in this complex of proteins, and we'll look at raptor, would sensitize to an ethanol exposure, but a mutation here, which then would cause an upregulation of this pathway's activity, should protect. And so we've tested those two possibilities, and they both prove to be true. So again, you have the control fish for raptor that have 2 good copies, 1 good copy or no good copies. And the focus right here is this heterozygote here. Again, this haploinsufficiency once you add alcohol that you don't see otherwise. And then, so we then took a look at this TSC1, this negative inhibitor, in whether or not it could rescue the effects of ethanol on these Pdgfra mutants. So the mutation of this gene itself causes no defect that we can observe. The fish are actually viable and they live to adulthood. But then if you add the mutation to a Pdgfra heterozygote or a mutant, you also fully rescue them. So this is, if you mutate this downstream inhibitor of mTOR, you can protect against the ethanol exposure of an upstream mutation. And so we were really excited about that. Let me go back, because mTOR is also known to be involved in social behavior. And in addition to facial defects that can be seen in children with FASD, there are very common behavioral deficits, and one of them being inappropriate social behavior. And zebrafish are useful for this because they're highly social. So that's a single zebrafish in a big tank. There are some other little zebrafish over here in this tank. Right now, I can't see them, but then Johann, my postdoc, removes that barrier. It can see them, and it spends all of its time over there. And it's a highly quantifiable behavior. And so these are little heat maps. I think that they're very useful. And then we can just measure the time that they spend in this little region closest to this tank of other fish. And so any normal zebrafish, when they're in this social behavior assay, will spend about 80% of their time in that zone. And that's an important number here in a little bit. If you expose them to ethanol for a very short window of time, you blunt that response, and it's really clearly evident in this heat map that you see here. So then we wanted to see, can mutating TSC1 restore this behavior in an ethanol-exposed fish? And so this is what you just saw, and I'm just showing you the heat maps right now. We'll get to the bars in a little bit, but now, here again, you just have 1 good copy and 1 bad copy of this gene, the same ethanol exposure, and you restore this behavior. And so it seems like the loss of TSC1 suppresses these defects. And again, remember this 80%. So here, this is what I just showed you, this heterozygote. So 1 good copy, 1 bad copy. They behave the same as controls. Mutants also behave the same as controls. The N here is a little low still, where this is ongoing research. But interestingly, now in the ethanol-treated wild type fish, they also are behaving at this 80%. And so we find this really fascinating. And one important thing about this analysis is that these fish here grew up in tanks with these fish. We don't genotype them until after we've done the analyses. And so we think that there's actually an environmental influence on these fish by growing up with fish then that are genetically resistant to these defects. And that's ongoing research that my postdoc is doing. So if there are just a few things that you take away from this, I would say one of the most important is that we see in gene-environment interactions, they can be synergistic. And so then the phenotypes that come from them are not readily predicted by the phenotypes that you see in just the genetic mutant or just the environmental exposure to a wild type. They can be very different. I didn't go into it, but mTOR is not the only mechanism. We know it is not the mechanism for this interaction. And so there are multiple mechanisms. And lastly, that gene function is contextual, and we need to keep that in mind, that under normal conditions, Pdgfra is only required for migration. It is not required for survival. But when you have an environmental exposure, now suddenly it is. So in this particular context, it is necessary for survival. And so then we're still working on a few questions. Really, we've only scratched the surface on what the genes are and what the mechanisms are. And we're really interested in expanding this to more chemicals beyond just ethanol. So these 85,000 chemicals. Can we start predicting interactions between multiple chemicals and gene environment interactions? So I'd like to end by thanking the people in the lab. You're going to see how the lab has evolved over the years here. I've had the pleasure of working with a great group of people. The lab has grown to where we can actually do for our lab photos, motion pictures as we start here. So these are the people currently in the lab, some of the alumni. We have great collaborators, and then of course the funding through various institutes at NIH. So thank you for your attention, and I hope you enjoyed the talk.